Hello everybody, my name is Eric Kelly. I'm an undergraduate student in Dr. Hasty's Biology 105L class. And today I'm going to be going over a really cool study that I found that came from a lab at UNC's Lineberger Cancer Center. So the study came from the Davis lab. They focus mainly on how genetics is related to cancer and how we treat certain types of cancer by using genetics. So in this study, they identified a type of rare bone cancer called Ewing sarcoma. It's most common in young adults and children. They found a certain vulnerability of the cancer, and then they used an artificial intelligence program to find a drug that would treat the cancer. So let's get into it. First, we need to say that cancer is a very tricky disease to treat because every cancer is different. There's no magic cure, and every type of cancer likely needs a different treatment. Part of this is because cancerous cells are almost just like your own cells, and we can't use a treatment that kills our own cells because it would kill us. So, the hard part is figuring out what makes the cancer different. How can we target only the cancerous cells and not all the rest of them? Let's take a look at the focus of the study. So, here's how Ewing sarcoma starts. One cell will have a mutation, and in this case, a piece of one chromosome gets swapped onto another. And what this does is it smashes two different genes together to create an entirely new gene. And the new gene just happens to cause cancer. The product of the new gene is a protein called EWSFLI1, and what it does is it acts like a go signal for the cell. It tells it to start dividing really, really fast, and so a large mass of these cells forms and it creates a tumor. So we have this new protein, and it's bad. We don't want it. But thankfully, there's another protein that destroys it, called SPOP. So we like SPOP, and we want more of it. However, there's another protein that destroys SPOP, and so the tumor keeps growing and our savior is gone. So as you can see, it's regulated. Everything in our body is regulated with chains of signals just like these. Now what we want to do is break the chain and get rid of our cancerous protein. There's two ways to do this now. We get rid of OTU D7A or make more SPOP. In this case, the researchers decided it would be better to just get rid of OTU-D7A, so they started looking for drugs that would bind to it and inactivate it. Now usually this would be an incredibly difficult process, and you'd have to test thousands of drugs by hand. But nowadays, we have artificial intelligence that can do it all for us in minutes or even seconds. So here's what they did. They told the AI what they were looking for. They knew the structure of the protein, and they told it to find drugs that would fit into this protein. It tested over 4 million molecules, and it came back with several hits. So they chose one that they thought would work well, and they named it 7AI. Now, to test their new experimental drug, they took mice and implanted them with tumors. One group of mice was given the drug, and another group was left untreated. After 25 days, they took out the tumors and examined them, and the results are pretty outstanding. As you can clearly see, the tumors on the left are much bigger than those on the right, which were treated with 7AI. They also weighed the tumors and tracked their growth over time. Now, not all of the varieties of Ewing sarcoma responded to the treatment, but for those that did, it worked very well. So now we know that the 7AI is capable of shrinking tumors or stopping them from growing. So where do we go from here? It's possible that it could be used in humans, but before that happens, there will need to be a whole lot of testing and safety checks. And perhaps they'll examine some other drugs or variants of 7AI to see if they can get it to work better. It's also possible that 7AI will be combined with other chemotherapies to treat Ewing sarcoma. All in all, the researchers were very thorough in their methods, and there's many aspects of the study that I didn't even show. So it's a really impressive discovery thanks to the scientists in the Davis lab, and potentially they will save lives. Here's the citation for the paper. Please check it out and give it a read if you're interested in knowing more. Thank you.